talk about DNA as the genetic material. Here's the structure of DNA. It's made up of 4 types of nucleotides. DNA is made up of a long chain of nucleotides. Each nucleotide has 3 parts. That's the phosphate group, which is designated by the circle here. The D-deoxyribose sugar, that's the D in DNA. And that's right here, the deoxyribose sugar. And then the nitrogen-containing base. So this will be your A, T, C, or G that we'll talk about here in a minute. Okay. All right, so here's the different nitrogen bases. We started working on this in class when you're doing your little coloring project. They could be either thymine, which is the T, cytosine, which is the C, or in our other category here called purines, these were called pyrimidines, these are called purines, we have adenine and guanine. What's the difference between the two? The pyrimidines, as you can see, just have a single ring, and the purines have a double ring. I don't know the best way to think about memorizing that. Maybe you can just think of a single pyramid standing there, and you're like, okay, the single pyramid, that's the pyrimidine, and the other one's the purine with the doubles. Also, the ones that match up together, that is, the ones that are complementary pairs, for example, in this case T and A, one's a pyrimidine and one's a purine. I don't know if that helps to remember this as well. Okay, so classically this structure of DNA, how it was first discovered, the people that have always been the most recognized have been Watson and Crick, and so they are given the most credit in regards to discovering the structure of DNA. But as we'll see, there's a little bit of scandal involved with this, a little scandalous behavior on the side of Watson and Crick. We'll get to that in a minute. So they're not totally deserving of all the credit themselves, but nonetheless, they did publish the paper, the one-page paper in the magazine Nature that described the structure of DNA. And that structure was described as a double helix. All right, and I think everybody, or a lot of people, are pretty familiar with that. But this is another person in this whole process that deserves a lot of credit. And her name is Rosalind Franklin. And she, pictured here next to her X-ray photograph, essentially what she took is a process called X-ray crystallography. She was able to photograph a molecule with an X-ray and sort of did provide this image. Maybe you can appreciate how this looks a little bit like an X-ray that you would see of a human skeleton. In this case, it's an X-ray of a molecule. And that is what sort of gave that shape that could be imagined by Watson and Crick as being a double helix. So she was often kind of pushed off to the side in this whole story, but now people are coming around to the fact that she played a big role in it. And then another gentleman, Erwin Chargaff, he is credited with coming up with the idea of the base pairing rules, that is A with T and C with G. And that's what we're going to talk about here briefly. And here is that structure, that double helix structure, that is a ladder that has been twisted. And A should always pair with T, C should always pair with G. And because of the pyrimidine pairing with the purine, the single ring with the double ring, we always have a uniform width. And here is a sort of an unraveled version of the double helix. You can see that what we call the backbone, that is all along here, that is connected by covalent bonds, meaning the electrons are being shared. And then here in the middle, the reason they're drawn as dots, sort of connecting the A with the T, the G with the C, T with the A, and so on, those dots are meant to represent something called a hydrogen bond, which we referred to at the beginning of the year briefly, or we alluded to. It's just a strong attraction. Basically, you have opposite charges on the sides of these molecules, and they are attracted to each other somewhat loosely. I mean, these bonds are not anywhere near as strong as a covalent bond. Okay, so this loose attraction, keeping them together, and you could imagine there's two here between these two nitrogen bases, whereas here there are three. So you might try to take a guess as to which one do you think will be stronger, the bond between the T and the A or between the G and the C.